Hello, everyone. For those of you who are just joining, we are going to start in just a couple of minutes, so please stay tuned.
And if we could have our first presenter go ahead and put their slides up so that they're ready to go when we're ready to start, that would be super. Thank you.
All right, well, my clock has 2 minutes after, so it's officially immunology time to start. So good morning, good afternoon, or good evening to you wherever you are located. My name is Rebecca Marsh, and it is my great pleasure to welcome you to this very special joint clinical case webinar hosted by both the Histiocyte Society and the Clinical Immunology Society. We are hosting this webinar today to foster relationships between our two societies as we both work to improve the outcomes of patients with HLH and other inflammatory inborn errors of immunity. As a member of both societies, I'm going to take just a couple of minutes to tell you about each of them. So the Histiocyte Society is a truly international organization with the mission to advance knowledge about histiocytic disorders and improve outcomes for patients. We are a small but mighty society with more than 200 physician and scientist members. Our members make genetic discoveries, implement new diagnostic tests, and conduct clinical and laboratory research in HLH disorders, including multi-center clinical trials. Our members have been instrumental leaders of several completed or ongoing clinical trials for HLH, including, of course, the HLH 94 and 2004 studies, as well as clinical trials of ATG, alemtuzumab, emapalumab, and ruxolitinib. Importantly, our annual meetings are always in a fantastic location, which is another reason to consider joining the Histiocyte Society. We were actually just in Athens a few weeks ago. The Clinical Immunology Society is a bigger, moderately sized society of about 800 members. We are located mostly in North America, but also around the world. We are devoted to fostering developments in the science and practice of clinical immunology. Our mission is to facilitate education, translational research, and promote excellence in the care of patients with immunologic disorders. We have a very vibrant membership consisting of clinicians, researchers, and leaders in the IEI field who are all actively working to advance the field. I would like to just highlight that our members benefit from a host of really excellent resources. We host several educational schools, including our flagship CIS summer school program, along with our diagnostic school, AIM program, and IOPIDS course. We have too many active committees to name, but we have tons of opportunities to get involved in advocacy, research, and education. And our best tool that we offer is a heavily used listserv where you can solicit expert opinions about patients. And we have a lively annual meeting that includes a fantastic talent show, and that's definitely our most popular event of the year. And with that, I hope you will consider becoming more involved in either the CIS or the Histiocyte Society. And I will now introduce our first speakers. So our first speakers come to us from the Histiocyte Society. It's my great pleasure to welcome Kai Leemberg and Kirsten Schultz from University Medical Center, Hamburg. And with that, I will turn it over to both of you.
Yeah, thank you so much, Rebecca. So I will start first, and then I will hand over to Kirsten Schultz. So our case is in an immune dysregulation syndrome with predisposition to HLH. And if you think that sounds boring, we'll prove otherwise. It will be a tricky case, and we are really hoping for your interactive participation in this. So we are really happy about this joint seminar. And when I prepared the slides, I put in the logos. And if you look at them, they're all in the same color. That seems to mean something. The Histiocyte Society, the Clinical Immunology Society, and the logo of our institution here in Hamburg. So it really does seem to mean something. Okay, so trying to move my slides. Right. So it is a couple of years ago that one of our younger physicians was on a rotation on the intensive care ward, and as she had been in contact with immunology for quite some time, so she came to us and said, well, there's this girl. She's about 5 or 6 weeks of age. And basically, she has had diarrhea since her first days of life. And everyone has been involved already, the geneticist and obviously the gastroenterologist. Could this be something that is of immunological origin, an immunodeficiency? So what do you think? What are your differentials? A child that was basically born with diarrhea, and you are asked as the immunologist, what are your differentials? What would you test for? Don't worry, this is just the warm-up question. So I hope I can see the chat. I can. Yeah, it is the one. IL-10. Okay, anyone else? IPEX. Excellent. IL-10 receptor. Perfect. Any other immunological diseases that are associated with enteropathy or with inflammatory bowel disease? ZIAP, yeah, obviously. It's a joint seminar with the Histiocyte Society and RC4. Okay, excellent. In fact, you mentioned very special ones. We thought of many of those. We did a checkup of the immune system to exclude combined immunodeficiencies, to obviously exclude IPEX, to exclude defects of the neutrophils. Even though for many of these conditions, it's obvious that usually inflammatory bowel disease appears later than that. But basically, the workup was mostly normal. Those serum immunoglobulins were normal. The CD4, CD8 counts were basically normal. The naive CD4 were normal, so it didn't look like a SCID defect. I will come back to the expansion of the effector CD8 T cells later. But all the other investigations of a general immunological workup returned normal. Basically, as an immunologist, I said, well, it is maybe not immunodeficiency, but she insisted and said, you remember there are some other defects that are very special. She said, so there are immunodeficiencies that are associated with HLH and enteropathy, and the ZIAP deficiency was mentioned. Do you know of any other HLH defect that is associated with diarrhea? Yeah, CGD and syntaxine binding protein were mentioned. In fact, CGD can be associated with HLH. Well, usually enteropathy or inflammatory bowel disease appears at a later stage, but it is quite true. And syntaxine binding protein is deficient in FHL5. And in fact, that was the condition that she was thinking of, because patients with severe FHL5 can have enteropathy from the first days of life. So when we think of the most frequent classical HLH defects, we usually think of the cytotoxicity defects that affect trafficking or that affect perforin directly as the effector molecules of the cytotoxic activity of CD8 T cells and of NK cells. So on the way of the secretory vesicles in the cytotoxic cells, A number of molecules are relevant to have that process properly performed. And one of these proteins doesn't work all right. So this process can't work properly, and the cytotoxic T cells can't exert their function, which in the typical HLH situation leads to a constant activation of these cells, of macrophages, of T cells, which leads to the full clinical picture of HLH. In this patient, there was no HLH whatsoever. So I said, well, this patient only has enteropathy. Why do you think of an HLH, a genetic defect? But she insisted and sent material to a lab which we cooperate with, and which is an immunological test that you perform if you want to exclude a cytotoxicity defect, if you don't want to go into genetics right away. What do you test for? Yeah, so I think this is CD107A, yeah, excellent. So it's the degranulation test. And in fact, this is what we did in this patient. Because my young colleague insisted, we did the degranulation test. And what you can see here on the left-hand side is the control. So you can see NK cells on the left-hand side. They are unstimulated, and when you stimulate them, you see a shift of the NK cells toward the right-hand side, which is CD107A, which is a marker of degranulation. And on the right-hand side, you can see the patient, and you can see that this shift of degranulation is much less. So we have this patient. She doesn't have any signs of HLH, but she does have this degranulation defect, though it is a moderate one. There are much severer ones. So in these cases, we still do targeted sequencing. So we like keeping our brain switched on and not switching them off, and only do exome sequencing. So we did targeted sequencing and found no defects in the classical genes for FHL3, 4, 5, and Griscelli syndrome type 2. But in fact, this patient did not only have the enteropathy, she had a number of other features. So we looked further if there were other defects of trafficking. We tested the platelets. If the secretion was abnormal, which in fact it was below the range, so that indicated that there might be true trafficking defect. And we tested the hair, which, as you know, in, for example, Griscelli syndrome, may be hypopigmented. And then the workup was going on. The geneticist, in fact, did an exome on top. The results weren't back yet. As you can see on the left-hand side, is the patient when she was 1 year of age. There were syndromic features. Other than that, there were problems endocrinological, neurological, and others. And the patient had a CMV viremia, and she did develop HLH. So at that point of time, we thought that can't be coincidence, that we have a cytotoxicity defect, that we have a partial form of HLH, though it was mild and went back to normal rather quickly, and we investigated this further. And what we did will be explained by Kerstin a little later. What the geneticist found was a defect in the MAP kinase activating death domain, which is a gene that we had never heard of previously. And I expect that no one of you had heard about it before. And we digged into this further. The neurologist got in contact with other working groups that worked on this gene, and in fact, 20 patients were identified that have this defect. And as you can see, all these patients are syndromic. Their facial expression is, well, classically with this myopathic open mouth faces. They have a high or broad forehead. The nasal bridge is typically depressed, and it's wide. The nose is short, and their face has a characteristical expression. However, there is a certain genophenotype correlation. So the full phenotype, which our patient had, has severe developmental delay, problems with the endocrine system, nervous system disorders, some hematological anomalies. Some do have fever of unknown origin, though only one other patient was formally determined to have experienced HLH. And there's the other group with a merely neurological phenotype, with some variable degree of developmental delay, some muscular hypotonia and speech impairments, but without the other complex syndromic features. So, and now our patients had most of these, in particular failure to thrive, hypoglycemia, anemia, and this partial HLH phenotype when triggered with CMV. And Kerstin will explain you now how we digged into this to, well, determine if there is a correlation between the HLH and the pathophysiology of this condition.
Yeah,
And
so
Professor Lemberg, maybe this is a good moment to pause. I think we've got a bunch of questions in the chat if y'all want to take a quick look at those and see if you can provide some comment.
So in fact, Chiari-Higashi syndrome was excluded by cytology. So usually, Chiari-Higashi can already be diagnosed in the blood smear and in the bone marrow as well. And the hair was macroscopically normal. In fact, this patient did not have EBV. The mortality in the entire cohort was mainly related to the endocrinological features, the neurological phenotypes, seizures, and mainly occurred in the first group with a full picture. Interesting questions regarding enteropathy. In fact, a number of these patients have constipation. Apparently, our patient was the one that was most affected by enteropathy. And for some patients, diarrhea is reported, but apparently it is not a universal feature. Well, Hermansky-Pudlak, well, obviously, all this was included in the exome, but given that the platelets were abnormal as well, that might have been a candidate. With Hermansky-Pudlak, usually the hypopigmentation is quite intense. Yeah. Right. Well, row G was excluded on the screen. Yeah. And regarding the NK killing, Kerstin will give you some more information.
all right, so I will hand over a little bit more to the, let's say, research part and background information about the disease or the gene actually or protein we found here in this patient. So as a short reminder, or maybe so to understand the mechanism, here's the cycle dependent on RAP27A that is necessary for exocytosis of the granula. So just briefly, RAP27A has an inactive and active form. So the inactive one is the GDP bound one, and there is activation by a protein, and then it will change to a GTP bound form, and then it can bind the vesicles, and combined with effector proteins, it can manage transport, docking, fusion of the vesicle, and thereby the exocytosis in the lymphocytes.
Du musst auf die Folie gucken.
Hier?
Ich glaube einfach, klick mal einfach auf die Folie.
Sorry. Okay. So here you can see a lot of interaction partners of RAP27A. So obviously there are more than one interaction partner, and depending on cell type and also, of course, of the protein they are interacting with, and RAP27A can promote different functions. So I think the most common one in literature is the melanophilin, who can manage together with RAP27A and also actin, myosin, the distribution of melanosomes and melanocytes. But there are also other ones, and I actually covered one here. And this is one protein that is the most, I would say, common one in context of HLH. So do you know what we are looking for here? Which protein interacts with RAP27A? Okay, I cannot see any comments for that. So yeah. So Claudio, that's right. So it's MUNC13-4. Or also called MUNC13D. So it's the gene for it. So yes, right. So we all know this, or most of you know this protein from the FHL3. So we're looking a little bit further. So when we think about HLH and syndromes or FHLs containing this, there is one that is a RAP27A deficiency, or it's a primary RAP27A deficiency. So it's the Griscelli syndrome type 2. So we basically have no RAP27A working in these cycles. And that's why we have this degranulation defect, but also have this pale hair, because there's no melanosome distribution and other features we can see in this syndrome. We also mentioned MUNC13-4, and this is actually an effector protein here at the docking tethering step. So when we have a mutation here in this gene, we have no interaction with the RAP27A, and thereby no docking step, and they're missing exocytosis in this case, and this also causes HLH, or better known as FLH3 in this. So question 2, are there other interaction partners whose dysfunction can cause an HLH? What do you think? Nothing? Yes, yes, of course. So I think it's quite obvious that any of these interaction partners that somehow influences the function of RAP27A can also influence, or in my opinion at least, can influence the cycle and thereby the exocytosis. And that's what we have with these MADD. So short, what is MADD? I think most of you don't know it, me either, before this case. So it's a quite large protein that is expressed in nearly all cell types, but there are different isoforms. So at least there are 7 whole length isoforms and also shorter isoforms. And what we know is there is an interaction of the TNF-alpha receptor, and it can propagate cell death or cell survival, depending on the isoform. And more interesting in this case, there's also an interaction with small GTPases, and especially for RAP27A and for RAP3. So MAT is actually the exchange factor of RAP27A, so it can activate by exchanging GDP to GTP, as I told you before. And our patient actually had this point mutation, what you can see on the top. It's located in the splice region of this exon, or behind the exon 4. And this actually leads to a deletion of a part of the protein. And what we could show, or the Schneeberger et al. Paper could show, is that this deletion leads to a reduction or even loss of protein inside of the cell. So when you go back to the cycle, this step here at the top, so the activation site maybe is infected by the mutation in our patient. So this is what Kai already showed you. This is a cytotoxicity assay. So we checked for degranulation in NK cells and also in T cells in our patient compared to healthy control. And what we can see is when you measure the degranulation marker CD107A, that there is nearly no degranulation in our patient. And this is true for the NK cells, but also for CD8 T cells and gamma delta T cells. We also checked for the killing capacity of the patient cells and simply measured the percentage of killing to a target cell line. Here it's K562, and we compared our patient to a Griscelli syndrome type 2 patient. And what you can see is that they both are quite similar. So both show nearly no killing toward this cell line. I just had a quick look to the chat. Do we check regularly for gamma delta T cell green?
No, that was an add-on.
Okay, so that was an add-on actually. So we had further investigation as we noticed that there are degranulation defects, and then we checked a lot more functions here in this patient. Right, so now we had a quite good hint that the MADD mutation in our patient could cause the degranulation defect and also the HLA symptoms, but there was not a real proof, because maybe we missed something else, other mutations, or I don't know. So that's why we created a CRISPR-Cas9 based knockout of the MADD in a cell line NK92, and performed the assays again. And that's what you can see here. Is also a degranulation assay. And after stimulation with K562 cell line, there was nearly no degranulation of the knockout cell line, and only with a quite strong stimulation of PMI Iona, we had a little degranulation, but it was far less than the wild type. And we also checked for killing capacity, and it was the same picture. So compared to wild type, the MADD-KO cell line had a impaired killing capacity. And that actually was the hint for us to show, okay, we would say MADD is the coursing gene or protein in this patient that leads to the degranulation defect and the at least partial HLH phenotype. So this is the mechanism we proposed that are going on here. So we saw this cycle before, and MADD is the exchange factor from GDP to GTP. And if MADD is missing, so that's the case in our patient, because of the mutation, we have no MADD protein in our cells, we have no activation of RAP27A, and thereby the transport and docking step is not working, and we have this degranulation defect. So question for you, would you call this FHL6 or maybe Griscelli syndrome type 4? Not sure. Why?
GS4.
GS4.
Has or normal? No
No,
organism.
no.
Both.
Maybe both.
Well, in fact, it's a really syndromic disease. So there are so many features, and the defect is only one of those. So there is the cytotoxic defect. And of these 20 patients, of this cohort, only one patient, our patient, we are aware of that she developed HLH. Maybe it wasn't looked for properly in the other ones. And we are aware of another one from Spain. So it is not one of the most prominent features.
What cause of early mortality?
Well, mainly endocrinology, hypoglycemias. So Scott suggests completely different names.
And Fiona asked for the interaction. So yes, actually MADD is the exchange factor of GDP to GTP for RAP27A. Other proteins involved, good questions. We don't know actually. So maybe.
Okay, so we are running 32 minutes. So it's the last question for you. So will you test any patient with HLH for this mutation? So I don't know what you usually do. Do you always run right from the beginning a large panel or an exome? In this case, yeah, obviously you should include MADD. But all these patients, or all the patients that we are aware of, do show some degree of syndromic features. So if you do have a patient that is compatible with these features, do test for it. But in an otherwise normal patient, it is rather unlikely that you will find an MADD mutation, unless some more hypomorphic mutations will come up.
Yeah. Two questions.
And I am going to actually cut us off here because we are 2 minutes over, and I want to make sure we have time for our next case. Hi, and Kristen, this was an amazing case. Thank you so much. We can keep answering questions in the chat, so feel free to keep offering up things and answer those there, but I will turn it over to Haney now. And Haney, you're still muted. Sorry.
I'm so sorry. Thank you, Rebecca, and thank you to our colleagues in the Histiocyte Society for that amazing case. We really enjoyed it. And good afternoon to those in a different time zone. Good morning in this time zone. My name is Hanadis Ale, and I go by Hani. I am a pediatric immunologist analogist at Joe DiMaggio Children's Hospital in Memorial Healthcare System, and I'm a proud member of the ECI committee of the CIS, and I will be your moderator for today's second case, which I hope you find it as interesting and challenging as the first one. Our presenter will be our first-year fellow in allergy and immunology, Dr. Srina Desai, and we are honored to have as our senior mentor, Dr. Dimana Dimitrova from the Center of Immuno-Oncology of the National Cancer Institute of the NIH, who has been very involved in this case and actually performed the patient's bone marrow transplant. Thank you, Dimana, for taking the time to join us and for being our mentor. I also would like to mention that I had the full support on this case of Dr. Gigi Notarangelo, who from the very beginning helped me with the final diagnosis and the management of this patient before the transplant. Gigi unfortunately could not be here today, but we are very grateful of his help. And without further ado, please, Srina, take it away. And I will be in the background monitoring the chat for questions and comments.
Hi, good morning, good afternoon, everyone. Let me just share my screen. All right, so my name is Srina Desai. I'm an allergy immunology fellow down at Memorial Healthcare System in Joe DiMaggio Children's Hospital in Hollywood, Florida. And I am very excited to present this case on a pediatric patient with RAG1 deficiency caused by novel variants. We'll be presenting this case through the lens of diagnosis to bone marrow transplant and immune reconstitution. So I have no disclosures. So let's dive right into the clinical presentation. So here we present a 4-year-old African-American boy who presented with recurrent otosinopulmonary infections since infancy. His infections were only temporarily alleviated with multiple courses of antibiotics, and he also presented with a recurrent pustular rash from the time he was 2 years old. In regards to more medical history, so he was also prematurely born at 30 weeks due to preeclampsia. He was diagnosed with hypothyroidism at 1 year of age, and he also had severe constipation with perirectal abscesses. So I'm going to pause here. What labs do we want to order?
So we have immunoglobulins, lymphocyte subsets with naive and memory T cells, a hemogram to begin with, IL-10 and IL-10 receptors, sequencing our IBD panel, workup for CUD, like a neutrophil oxidative burst. Great, CD62L, shedding, FOXP3. Wow, we got a lot of labs here.
Awesome. So many of those labs we did order. So in regards to his immune function, here I have his initial immune function at presentation, which was in January of 2018. So to quickly summarize, all of the abnormalities are highlighted in red. So for the most part, everything was low with the exception of his NK cells, which were elevated. So to summarize, he did have hypogammaglobulinemia. He had decreased CD19 positive B cells. He did have a normal T cell count. He had increased NK cells. And his functional antibody titers to antigens were non-protective except for anti-rubella and anti-tetanus. And his total complement neutrophil oxidative burst assay and lymphocyte mitogen and antigen proliferation tests were normal. So we did not find an alternative explanation for the hypogammaglobulinemia, such as a protein loss enteropathy. And the increased NK cells is likely compensatory since they don't have T and B cell receptors. And I also want to note that the patient did have positive anti-thyroid antibodies, although his hypothyroidism did self-resolve. So I know our title gives it away a little bit, but given the labs, what other diagnoses come to mind without knowing this was a RAG1 deficiency?
So X-linked agammaglobulinemia was the actual first diagnosis that came to mind when we saw the immunoglobulins because they were impressive, as Rebecca mentioned. But his BTK function was normal, so there was a lot of puzzling thoughts there. Icaros, nice, Cecilia, Icaros. Yep. And a combined immune deficiency, of course. CID, okay. CVID, Raul, very good.
So in terms of his diagnosis, we did a primary immunodeficiency panel, which identified 2 novel variants in the RAG1 gene. The NIH assisted with some in-depth T cell studies that were done by Dr. Notarangelo, and they included RAG expression studies, which varies based on the mutation and also correlates with disease severity. The flow cytometric analysis was used to quantify the T cell receptor, specifically the V alpha 7.2 chain expressing T lymphocytes, which revealed the patient only had 0.5% of T cells expressing this V alpha 7.2 chain. Other testing unveiled the patient's CD4 cells were skewed to TH1 and T follicular helper cells were skewed to TFH1 cells. And with the collaboration of the NIH, we were able to confirm the suspected hypomorphic RAG1 deficiency, which was induced by 2 newly identified pathogenic RAG1 variants. So more specifically, the flow analysis was used to quantify that T cell receptor V alpha 7.2 chain, which, as I mentioned, he only had 0.5% of T cells expressing that chain. Values in normal control are actually 2 to 8%. So if you take a look at the middle box in the bottom row, right around here, you will see that in our patient, there is no expression of this chain in the small box, which is compared to the control up here, which is the CD3 cells expressing this V alpha 7.2 chain. So we can conclude that he has an aberrant T cell receptor. And this is a strong indication that there is a pathogenic RAG variant. Patients with RAG deficiency are severely impaired in their capacity to rearrange distal V alpha genes. And specifically, these T cell receptors have the alpha and beta chains. So we did do further analysis of the V beta repertoire by flow, and it did not disclose major abnormalities, but again, the alpha did. So further testing also revealed the patient's CD4 cells were skewed to TH1 and the TFH or T follicular helper cells were skewed to TFH1. And these are bad T follicular helper cells. So the best cells are the TFH type 2 and the TFH type 1 are not. So this is, again, consistent with an aberrant oligoclonal T cell that make it out of the thymus. And this is not normal because you're not supposed to have only one lineage of lymphocytes. And so these T follicular helper cells are much more abundant than they should be. And figure 3 here shows that a high proportion of the CD8 cells were CD57 positive. And this suggests the presence of cytotoxic activated CD8 cells, which typically are poised to express interferon gamma. And the clinical importance of this is in the production of anti-interferon antibodies, which our patient actually did develop as well. So RAG1, so you need RAG1 and RAG2 to create T and B cell receptors. It stands for recombination activating gene 1. And normally this gene is involved with providing instructions for making a member of a group of proteins called the RAG complex. And this is involved in the process of VDJ recombination on T cell receptors. So during early B and T cell maturation, this recombination occurs in the bone marrow for B cells or in the thymus for T cells. And so far, there has been over 70 mutations in the RAG1 gene that have been found, specifically to Omen syndrome, which is a type of severe combined immunodeficiency. So with VDJ recombination, the variety of proteins produced throughout life following VDJ recombination provides greater recognition of antigens, and it allows the body to fight infection efficiently. The T cell receptor is a protein complex found on the surface of T cells that is responsible for recognizing antigens presented by the MHC. And the area of the receptor that binds to the antigens is the part undergoing that VDJ recombination, and that is for enhanced specificity and diversity of antigen recognition. So the RAG1 and RAG2 genes encode for specific proteins that play a crucial role in the early stages of T and B cell development, and they specifically initiate the recombination of variable diversity in the joining genes at that T cell receptor. So just to go through some step-by-step sequential events during VDJ recombination. Oh, it seems that maybe the images didn't load correctly. But first, there's basically synapsis and cleavage of the DNA at the heptamer or coding segment, and that's mediated by the RAG1 and RAG2 genes. And then there is the coding end hairpin, in which there is endonuclease Artemis action. And then these broken ends are repaired by non-homologous end joining machinery that's present in all cells. There we go. So in regards to RAG1 mutations, changes can impair the RAG complex formation and function, including its ability to bind DNA. And so when there is a mutation present, that VDJ recombination is diminished, and there is limited diversity in the proteins on the surface of B cells and T cells. And so mutations in RAG1 accounts for approximately 10% of all SCID patients. And RAG1 and RAG2 deficiencies also associated with abnormal thymic architecture, impaired thymic epithelial cell maturation, defective air expression, and reduced Treg production. There's a lot on this slide, and I'm just going to briefly go through it, but these are the previously described RAG1 deficient disease presentations. So far, there's been described a complete RAG1 deficiency, Omen syndrome, an atypical Omen syndrome, atypical SCID with delta gamma T cell expansion, as well as a delayed onset form. And the CIDG-AI is used to summarize the presentation of patients with combined immunodeficiency, plus or minus granulomas or autoimmunity. So our patient specifically has hypomorphic RAG1 deficiency, and this variant has not been previously described. He has two novel variants in the RAG1 gene, and this means that he has some residual VDJ recombination. So here's his lymphocyte subset trajectory. This is the labs that we have over time from his presentation in 2018 all the way through 2023. And they basically demonstrate decline in lymphocyte counts over time, which is a risk factor for developing autoimmunity and a contributor to the decision for us to ultimately transplant. So the numbers in red are below range. And as you can see, there are virtually no B cells. So now in regards to his treatment and transplant course, so he was initially initiated on IVIG as soon as we found out he was hypogammaglobulinemic in 2019. In January 2023, the decision was made for him to receive bone marrow transplant. So he was admitted to the NIH and he received a reduced intensity conditioning haploidentical T-replete bone marrow graft. His donor was his brother, who also did require RAG mutation testing. And the treatment of the regimen is outlined above. So he received a combination of pentostatin, low-dose cyclophosphamide, and busulfan. In regards to graft versus host disease prophylaxis after transplant, he did receive high-dose cyclophosphamide, mycophenolate mofetil, and sirolimus. And post-transplant, he did require pentamidine for PJP prophylaxis, as well as other infectious disease prophylaxis. So in regards to his post-transplant course, he did have several immune and infectious complications, including low-grade hemolysis, EBV and CMV reactivations, as well as multiple bacteremias. He did receive his first IVIG 4 months after transplant and has received a few more IVIGs at our institution since then. He had consistently low IgA and IgM, but his IgG has been within normal range. And he did have B-cell reconstitution post-transplant in June 2023. Now, in regards to the infectious complications, these patients do produce anti-interferon antibodies, which does place them at greater risk for severe prolonged viral infections, which our patient did demonstrate. And de novo autoimmunity has also been noted in some of these patients post-transplant. So he did have successful engraftment with 100% donor T and myeloid cells in the peripheral blood. There's no graft versus host disease treatment at this time. In approximately 6 months post-transplant, he did show signs of recovery, evidenced by resolving complications and immune reconstitution. His IVIG infusion intervals have slowly been extended to every 8 to 12 weeks, which shows a really positive trajectory in his immune recovery with encouraging lymphocyte counts and detectable class-switched memory B cells. And he has not been on any immunosuppressants or infectious disease prophylaxis for many, many months now. He was discharged home and he remains under the care of his primary immunologist and bone marrow transplant physicians, as well as the guidance of the NIH team. This is his immune reconstitution post bone marrow transplant, and this is continuing to evolve. The bolded numbers show improved numbers, and the numbers that are in red are, although they're low, they're improving 6 months post bone marrow transplant. And he's now making IgA and IgM, which is new post-transplant as well. So this patient's journey has been marked by novel genetic discovery, state-of-the-art bone marrow transplant, and obviously demonstrates meticulous post-transplant management. It's a multifaceted case. I was very excited to learn all about it from diagnosis all the way to current state. And it's very exciting to see where he started and where he is now. It obviously elucidates the complexities involved in diagnosing and managing these really rare inborn errors of immunity, and highlights the importance of collaboration and a multidisciplinary approach for optimal patient care. So I just want to acknowledge some really important people who were involved in our little guy's care. Dr. Notarangelo from the NIH, who helped with a lot of the testing and diagnosis. Dr. Dimitrova, who we will hear from in a minute, who is our patient's transplant doctor, and Dr. Galvez, who was the head of transplant at Nicholas Children's Hospital in Miami, Florida, who supported Dr. Ale during his post-transplant course. These are my references, and I want to thank you so much for allowing us to present this case. I'm going to pass it over to Dr. Dimitrova, who will present some more information on a larger study done in these patients.
Great job, Trina. So before we get into the specifics of the study, I think it's important to point out that there's several really good learning points here. So this was a patient who arguably had relatively non-severe disease manifestations, but on testing, clearly he had profound humoral immunodeficiency. So getting started promptly on IVIG was critical to prevent further and more serious infections. And then additionally here, understanding or making the genetic diagnosis promptly was really key to understanding the fact that he actually had a combined immunodeficiency with T-cell dysregulation and risk for additional complications and potentially the need to be referred for transplant. And so that brings us to how we think about whether a patient needs to proceed with transplant and what is the optimal time. So like Shrina mentioned, we saw that his lymphocyte counts were decreasing over time, so that heralds further reduction in the T-cell repertoire and potential development of clinically significant autoimmunity. And we also know that furthermore, patients with hypomorphic RAG deficiency are at risk for destructive granulomas. So it's really important to find that balance of determining whether the disease manifestations are severe enough to warrant transplant, but also not waiting so long that the patient is no longer clinically well in a way that they can tolerate transplant. So really importantly, this patient going into transplant was in fantastic shape. He had normal organ function. He had no, bronchiectasis. His lung function tests were normal. Heart function was normal. Liver, kidney, all looked great. And in just the general transplant setting, going into transplant stable without active infection and with good organ function is associated with lower transplant-related mortality. And the importance of going to transplant earlier was borne out in this study specifically of RAG-deficient patients with a combined immunodeficiency phenotype. These were 60 patients described by Dr. Schutz in a paper that was published in Blood this year. And so you can see that there was a wide range in terms of the age from newborns to adults. Many or most of these patients had some kind of autoimmunity. About a third had active infection and more than half had organ dysfunction. And so you can go to the next slide. Shrina, are you controlling it or?
Yes, let me see if I can. It wasn't working. There we go.
Okay. And so, so just like we know that in the general transplant setting, earlier transplant before organ comorbidities have occurred is associated with better outcomes. We see this also in this group of patients where survival was poor in patients with organ damage going into transplant or with active infections. And more specifically in this cohort, the T-cell depletion of the graft and organ damage were associated with reduced overall survival in multivariable analyses. So next. And so what I find really interesting about specifically this group of patients is that patients transplanted later in life or after having had complications like infection, autoimmunity, organ damage, not only had poor survival, but they also had poor immune reconstitution. And they had de novo autoimmunity, which can sometimes be seen after transplant, but seem particularly prominent in this cohort. And so, something needs to be said about the role of the thymic milieu and the defective maturation of thymic epithelial cells and how what that might portend for long-term outcomes in these patients. Now, luckily, we do test various autoantibodies in all our RAG-deficient patients. And as we discussed earlier in the presentation, also in the chat, this child had anti-type 1 interferon antibodies, and he also had anti-thyroid antibodies, which declined over time and on recheck after transplant. Now he's not even yet a year out, so we don't have that much follow-up on him, but in our other RAC deficient patients, we have seen both more standard autoantibodies as well as the anti-cytokine antibodies decline over time, albeit slowly. That is all I have, but here, I'll pull up the chat, check if there's any questions. So this time frame for humoral immune reconstitution is very typical just in general in the post-transplant setting. It's pretty typical. I don't think we tested IL-6 antibodies. So I don't know that he really had a propensity for soft tissue and pustular infections. I don't think the rashes that he had were ever determined to be a specific infection. The perirectal abscess, he has a history of severe constipation. So, he hasn't had a recurrence of these issues since having a better bowel regimen.
And Damana, if you want to take one more quick question, and then I'm going to close this out because we are right at noon.
Sure.
Or maybe you got them all. I can't tell. I'm good at reading the chat.
So he did not have any granulomatous inflammation, but what we've seen in other patients is that this tends to develop later, like in late adolescence to adulthood more commonly.
All right, so I think I'll end this here. Again, if you enjoyed today, please consider joining or just becoming more involved in either the Histiocyte Society or the CIS or both. I want to thank again Kai, Kirsten, Shrina, Amy, Damana. This was wonderful. I really appreciate everyone's participation in the audience as well. Thank you to everyone, and we will see you on our next webinar. Have great days today. Guys, that was great. You guys knocked it out of the park today. So thank you so much. And Sawyer, good job running it. Good job enabling the chat. Everything went really smooth.
It's an excellent format. A lot of interaction. Thank you.
No, it's really lively. You could tell in the beginning. I noticed it was like less than usual. So I'm glad. Mike Jordan texted me and said he couldn't do the chat. So that was good. Yeah, and I agree. Maybe we should do this once a year in the fall. This would be a fun little thing to do. Yeah.
It is demanding, like the presentation and the chat and the control of Zoom, but it's a good format. Yeah.
I don't know if you guys noticed. I saw at max was like around 116 to like 120. I think the highest number I saw was 116, but I also wasn't looking the whole time, so could have been higher.
Oh,
that's pretty decent attendance.
Yeah, that's cool.
Yeah, it really is. Well, you guys have good days too, and thank you again. Really appreciate it.
Thank you. Thank you.
Thank you, everybody. Bye.
Bye-bye.